Hey there, how's it going? So we're back, and we're going to start talking about the next section of the Bio 101 course. And this section has to do with genetics and medicine, and so we're going to touch on a lot of things that many of you are probably familiar with. We're going to start by talking about cellular division and DNA synthesis. So I want to talk very briefly about the cell cycle. Then we're going to talk about the structure of DNA. We've already talked a little bit about the structure of DNA. We're going to go into a little bit more detail. We'll talk about DNA replication, making copies of DNA. And then we're going to talk a little bit about cancer. And so that, is a disease that is very common and it's related to these other topics. So let's get started. Okay, so first general question for you to ask yourselves. About how many cells do you have in your body? 30,000, 30 million, 30 billion, or 30 trillion? What do you think? Well, the answer is 30 trillion. Now, this is an estimate, and I see different estimates, but I found a reliable source that suggested this is a typical number of cells. 30 trillion. Can you even conceive of a number that big? You really can't. And what's interesting is almost every one of these has an exact copy of each of your chromosomes. And so you've got some cells that only have half your chromosomes, but 30 trillion cells, and they all have an exact copy of your DNA. That's pretty cool. Now, as a side note, you might have an extra 40 trillion bacterial cells living inside you and living inside your gut, right? Of course, those bacterial cells are much smaller than your cells. So on a cell-by-cell basis, you might be more bacteria than human, right? If you just counted the cells. Now, it depends upon how long since you pooped, right? You poop out a whole lot of bacterial cells every time you poop. So again, these numbers are just mind-blowing and really interesting. And to me, what's even more interesting is that every one of us started off as a single cell, right? When that sperm hit that egg, it was one cell. And then that cell divided and became two cells. And they each divided and became 4 cells, then 8, then 16, and now 30 trillion. And so again, that's, to me, that's just a fascinating fact about all of us, is that we all started off as a single cell. And again, I'm fascinated by the idea that the 30 trillion that eventually resulted from that all have the same DNA in every one of them. That's really cool. So let's kind of talk about that process. So now every time a cell divides to make 2 new cells, it goes through the same steps. But like I said, I said most of your cells have all your DNA, but you also have gametes that only have half the DNA. And so you've got kind of 2 types of cell division. You've got mitosis, which is cellular division of somatic cells, or body cells, or non-sex cells. And so in mitosis, you start with a single cell and you end up with 2 identical daughter cells. And so that's how you can go from, all of us starting as a single cell and then become 2 and then become 4 and then become 8. Through mitosis, you start with that cell and you just make an exact copy every time, or you make 2 identical daughter cells every time. Then meiosis is when you create the gametes, or the sex cells. So meiosis is the process where you create an egg or a sperm. And so the difference here, there's a couple of differences between meiosis and mitosis. In meiosis, you start with a single cell and you end up with 4 cells. And those 4 cells are not identical. And so that's kind of a big difference between meiosis and mitosis. In mitosis, you end up with 2 identical daughter cells. In meiosis, you end up with 4 non-identical cells. And each one of those cells is a gamete. And the number of chromosomes in those 4 cells is half the typical number. We call that haploid. So you start with a diploid cell, you end up with 4 haploid cells, and they're not identical. Now, the importance of this is, those gametes are going to presumably fuse with a different gamete. And so you have to have the number of chromosomes. You have to take the number of chromosomes and cut them in half so that when 2 gametes join together, then you restore the typical number of chromosomes. If you didn't do this, then every time 2 gametes came together, you double the number of chromosomes, and that would become unwieldy very quickly. So what's a way to remember the difference between these two? One way is mitosis has an I and a T in it. IT, identical twins. Mitosis gives you 2 identical cells. Meiosis has an E and an S in it. Eggs and sperm. Meiosis makes the gametes, the egg and sperm. So that's one way to try to remember which is which. So again, why do we have to have 2 different types of cellular division? If you're growing, you need to make new cells, right? So if you start as a single cell and you end up as a grown adult, you have to have more cells just to get bigger. And also, you're constantly, cells are dying, you're losing cells, you have to replace old cells. So you're going to need mitosis. That's just going to take an existing cell and end up with 2 identical cells, right? And so, that existing cell is going to get larger and then split into 2 small cells. And then each of those cells are going to get larger and then they're going to split into 2 small cells. So, the cells have to get larger in volume before they split. And each of those cells is diploid, meaning it's got the regular or the typical number of chromosomes. But if you're going to make the sex cells or the gametes, you need meiosis. They need half the number of chromosomes. They need to be haploid. That way, when 2 gametes come together to form a new organism, you get back to the diploid number of chromosomes. So here's a figure from your book, just kind of illustrating, the basic idea in mitosis and splitting the DNA between these 2 cells so that you can have identical copies. So again, it's one of the fascinating things about biology is that multicellular organisms, every cell has a full copy of all the DNA. But to do that, that means that you've got to make a copy of the DNA before you split the cell, and that's what they're showing you here. So in interphase, you make a copy of all the chromosomes. Now for a split second, for a few minutes, that cell has twice as much DNA as it typically has, but then when that cell splits, then each cell has the regular diploid amount of DNA. And so here's figure 6.9 showing you the steps of mitosis. And so you start with what's known as interphase. Interphase is not mitosis. That's just when the cell is growing, making copies of DNA. Then mitosis involves the same steps in the same order every time. Prophase, metaphase, anaphase, telophase. And then cytokinesis, cyto meaning cell, kinesis meaning cut or split. Cytokinesis is when then that cell splits into 2 new cells. So after that, after cytokinesis, then we're back into interphase, and each of those cells goes through this process again. We're not going to get into too many details about what happens during each phase. It's nice to know the phases and the order they come in. And so the mnemonic device we use is IP more after this Coke. Interphase, prophase, metaphase, anaphase, telophase, cytokinesis. And so That's good for you to know. I'll be honest, I bring this up and I want you to know this because I invented this. And I stand by that. There's lots of these mnemonic devices that, we all learned when we went to school, and who knows who invented them. But I swear that I invented this one. I don't remember being taught it. I can't find it anywhere else. I want credit for it. So this is my mnemonic device. So anyway, I pee more after this Coke. That's how you remember the steps of mitosis in order, right? That's my contribution to biology. Anyway, that's why I'm making a big deal out of this. Now, to get back to what we were talking about, a very important thing that happens during interphase is that copying of the DNA, or DNA replication, or DNA synthesis. Okay? That's a big deal, because that's, again, what allows every new cell to have the full copy of all the chromosomes. And so we want to talk about that in a little bit more detail. And of course, that is what I just said here. The take-home message is every time a cell divides, you must copy the DNA. So every cell then has the exact same DNA in it. So before we talk about copying the DNA or DNA replication, we need to talk in a little bit more detail about what DNA looks like. So let's talk about DNA structure. Okay, so a little, again, side note, history note. So you know that DNA is the source of inheritance, right? Even if you're not a biologist, you've heard the term DNA. You know DNA has to do with genetics and chromosomes, and that has to do with passing traits from one generation to the next. You know that DNA is important to that. And you know that DNA makes RNA, makes protein, because we learned that earlier, right? We know that that's common to all living things. And you know that a gene is a piece of a chromosome that codes for a protein. And so you've got a chromosome that has many genes on it. Well, what is a gene? A gene is a piece of a DNA that codes for RNA, which has the instructions for making protein. So you know all these things because we've been talking about them. But we haven't always known that. Back in the day, when scientists were first starting to understand the cell and learn about these different types of molecules, they could tell that, DNA, the cells contain DNA and they contain protein, but they didn't know how all that related to one another. And so I'm talking like early 20th century, early 1900s is when we started to really learn about cell biology. And so scientists could tell that these things had a lot to do with the characteristics of an organism and had a lot to do with passing traits, but they didn't know how it worked, and they didn't know if DNA or protein was the source of inheritance, right? And so this is part of the scientific discovery process. But over time, we were able to figure out that, okay, so DNA is the source of inheritance. And so that makes DNA incredibly important to living things because it is what controls the traits, and it's how the traits get passed from one generation to the next. And so once, again, in the early 1900s, that we figured out that DNA is very important, then it became important to understand, well, how exactly does that DNA work? And the key to that is understanding its molecular structure. So that's why this is a big deal. And so historically, that's why this was a big deal. And so that was a lot of work in the mid-1900s, is trying to figure out the structure of DNA, which would then unlock more of its secrets. And the names that are most familiar with this are Watson and Crick. They won the Nobel Prize because they published a paper describing this double helix, this structure of DNA that we've already talked about. Now, something that we like to point out is that they based a lot of their work on data collected by a woman named Rosalind Franklin. And we like to point her out because she kind of got shafted. She does not get a lot of the credit for discovering the structure of DNA. But Watson and Crick used a lot of her work to figure out and write their paper. Now, that's how science works, right? We talk about scientists learn from other scientists and build upon their work, but she was really important to understanding the structure of DNA, and we'd like to see her get a little bit more credit. Now, what she did and what Watson and Crick used was X-ray crystallography. And that's a technique where you take a molecule and you shoot X-rays into it, and then they develop, some film, and by looking at the patterns that are created on this film, you can infer the shape of a molecule in this way. And so from that, they were able to all figure out, Watson and Crick were able to figure out the double helix shape of the molecule based upon this, one thing they used was Rosalind Franklin's X-ray crystallography. And so, what is the structure of DNA? We said it's a double helix, but let's talk about it in more detail. The monomers that make up nucleic acids like DNA and RNA are called nucleotides. And so those are the building blocks of the nucleic acid. So just like, the building block of a carbohydrate is a simple sugar. The building block of a protein is an amino acid. The building block of a fat is a triglyceride and a hydrocarbon. The building block of a nucleic acid is a nucleotide. Now, in DNA, there's only 4 of these. There's only 4 that's used to make any DNA molecule. And so that's pretty cool, right? With 4 nucleotides, you can make any DNA molecule, which means you can make any organism. And so those 4 are adenine, guanine, cytosine, and thymine. A, T, G, and C. Adenine, thymine, guanine, cytosine is the order I have here. So to think about it this way, there's only 4 letters in the DNA alphabet. And all information in DNA can be spelled using those 4 letters or those 4 nucleotides. And so this is just kind of give you a little bit of an idea. This is not the whole nucleotide here. This is just the nitrogenous base. We're not going to get into too many details, but a nucleotide is a pentose sugar, a 5-carbon sugar, a phosphate, and then one of these nitrogenous bases. And so it's the nitrogenous base that determines which nucleotide you have. And again, just to put this in context, this is a figure we saw earlier. DNA is a type of molecule. A short piece of DNA that codes for a protein is called a gene. And a very long piece of DNA with many genes on it is a chromosome. So just kind of put these terms all in relation to one another. Oops, excuse me. So DNA is double-stranded. And so it's got 2 strands, and each is a long string of nucleotides. And then those 2 strings or those 2 strands come together. And so when those strands come together, they don't come together at random. The base pairs, the nucleotides on each strand, pair up in a very specific way. Adenine always pairs with thymine. And cytosine always pairs with guanine. And so the way you remember which pairs with which is all tigers can growl. AT goes together and C and G goes together. And so you say, well, so what? Well, this specific biochemical relationship is huge and is incredibly important and means, it's kind of important to this whole replication process. So first off, let's look at this structure again. If you zoom in on the strands that we were showing here, if you zoom in on a piece of DNA, you can see that they're trying to show those nucleotide bases pairing up. And C is always with G and A is always with T. And so that's called a base pair. And so you get lots and lots and lots of these base pairs, an incredibly long strand of them. That's a DNA molecule, that's a chromosome. And so humans, our total chromosomes is about 3 billion base pairs. So you got 3 billion pairs of these nucleotides in every single cell. That's incredible. But here's why it's so important that those nucleotides pair up in the same way every time. That means if you only have one strand of DNA, you can automatically figure out the other strand, which is known as the complementary strand. Remember, DNA has 2 strands. And so if you were to pull those strands apart and you only had one strand, you could still you could repair that DNA, you could build another strand because you know how the base pairs pair up. And so it's such a simple biochemical relationship, but it has huge implications. And so I think it's fascinating. And so when you're copying DNA during cell division, you can use this fact to make new copies of DNA. So remember back, we've been talking about cell division, right? We started talking about mitosis, and we said during interphase, you got to make copies of all the chromosomes so that when the cell divides, it each gets its own complement of DNA. Well, how can you make an exact copy of DNA? Because you know how these base pairs pair up. So that's awesome. And so, for example, here's a short strand of a DNA molecule. Remember, humans, you got 3 billion of these, and here we're looking at 5. So this is just a short strand for experimental purposes or example purposes. So if I only had one strand, that's not enough, right? DNA needs 2 strands. Well, what's the other strand? Well, I see ACTAG, I know it pairs with each of those. And so for any single strand of DNA, excuse me, you can always figure out the complementary strand. So A must pair with T, C must pair with G, T must pair with A must pair with T, G must pair with C. So that's how you can build a new strand from an existing strand. And so here, just practice. What's the complementary strand for this piece of DNA? Well, you see the first base pair is A, so its complement must be T. And so the next one must also be T, the next one must also be G. So pretty quickly you can find that this last choice would be the complementary strand. If you know one strand, you can figure out the other strand. All tigers can growl. And so once you have those 2 strands and those nucleotides are holding together, that looks like a ladder. And if you take that ladder and you twist it, that's the double helix, and that's the structure of DNA. And so that's what I'm trying to show you here. And so you can see, you can kind of imagine how you got this sort of twisted ladder, and the rungs, the middle part that you would step on if you were on the ladder, those are the nitrogenous bases. The sides of the ladder that make the frame of it, that's the sugar phosphate of those nucleotides. But more importantly, if you look here, you can see how those nucleotides are pairing up. A is always with T and C is always with G. Okay, so that's the first part. And so then in the next lecture, in the next PowerPoint, we're going to talk a little bit more about DNA replication. So I guess I'll see you then.